Today we are going to talk about one of the very important function of the cell membrane. As we have discussed previously, that cell membrane is made of lipid bilayer with associated proteins, and it acts as a semi-permeable membrane. Now let's suppose here is a cell, and let's suppose here is a cell and this is its cell membrane. Now all those substances which are required by the cell, they need to cross the membrane and enter into cell. For example, oxygen need to be transported in, right? Amino acids need to be transported in, glucose needs to be transported in, even some hormones like thyroid hormone, they do enter into cell, and many other substances, they need to enter into cell, right? It means they need to be transported from extracellular environment to the intracellular environment. At the same time, many cells synthesize many substances. For example, if this is a glandular cell, it will synthesize some hormone, right, or some secretions, and those secretions which are produced in the cell, they need to be secreted out, or they need to be transported from inside the cell to the outside the cell. So what I am talking about, that all those substances which are required by the cells, they need to be transported from extracellular environment to the intracellular environment, and cells synthesize and secrete many substances or release waste metabolic products, they need to be transported outside the cell. And one of the major functions of the cell membrane is that it controls the transport of different molecules and substances across the cell membrane. Let's see what are the different transport mechanisms which are used by the different substances to cross the cell membrane. We will start with a very simple mechanism and gradually we go to the more and more complex transport mechanism. The simplest mechanism for the substances to move across the membrane is simple diffusion. That is simple diffusion. Now this concept is very simple. Everyone knows that substances diffuse from high concentration to the low concentration. So if there is a substance which need to be transported, let's suppose these are the molecules of oxygen, right? As you know, oxygen is constantly utilized within the cells. So it means oxygen concentration within the cell will be low, but in extracellular environment, oxygen concentration is high. So it means oxygen need to be transported from extracellular environment to the intracellular environment. But because oxygen is soluble within the lipid bilayer, because oxygen can dissolve into cell membrane, so it can easily transfer across the membrane, right? By simply oxygen from higher concentration is moving towards the low concentration, and cell membrane does not produces lot of resistance because oxygen molecule are very small and they are lipid soluble and they will dissolve into lipid bilayer, right? For transfer of oxygen from extracellular environment to the intracellular environment, we don't need any special mechanism. Then you know, when cells are metabolically active, they are producing carbon dioxide. So carbon dioxide concentration is high within the cell, right? Carbon dioxide concentration is high within the cell and it is low outside the cell. It means carbon dioxide need to be transported from the intracellular environment to the extracellular environment. Again, you know, carbon dioxide is soluble within the lipid bilayer because carbon dioxide can dissolve into membrane. So from high concentration, it can move to the low concentration outside the cell without much resistance. So such substances like oxygen or carbon dioxide or other lipid soluble molecules or very small molecules which can easily dissolve into cell membrane, they can move across the membrane in a simple diffusion method. Is that right? So this is the simplest you can say transport mechanism across the membrane, which is called simple diffusion. What is simple diffusion? Diffusion is the mechanism in which molecules move from high concentration to the low concentration, and which substances can easily diffuse across the membrane? Those substances which are very small molecular weight or which are highly lipid soluble. Is that right? So this is basic about the diffusion. Now diffusion depends, the rate of diffusion depends on which factors? What are the factors which determine that how fast a substance will be diffusing from across the membrane? Number one, rate of diffusion. For example, the rate of diffusion is directly proportional, number one, to the concentration gradient. If concentration gradient across the membrane is very high, substances will diffuse more rapidly. But if concentration of a substance across the membrane is similar, right, diffusion will be very slow. Is that right? So number one factor which determines the rate of diffusion is your concentration gradient. Then another mechanism, another factor which determines the diffusion is, yes, who will tell me? Surface area of the membrane. If surface area of the membrane is very big, diffusion will be easy and fast. But if surface area of the membrane is less, diffusion will be slow. So surface area also determines the diffusion. Then another factor which determines the diffusion of a substance across a membrane is solubility of substance within the cell membrane. Substances which are more lipid soluble, they will be diffusing more efficiently, and substances which are less lipid soluble, they will be diffusing less efficiently. So another factor is solubility of a given substance within the lipid bilayer, right? So we can say rate of diffusion of a particular substance across the membrane is directly proportional to the concentration gradient, directly proportional to the surface area of the membrane available for diffusion, and directly proportional to the solubility of substance in the lipid bilayer. But it is inversely proportional to the thickness of the membrane. If membrane become more and more thick, then diffusion rate will become slow. And secondly, this is also inversely proportional to the molecular weight of a substance. Of course, as molecular weight of substance increases, then rate of diffusion across the membrane will become less, right? So these were few words about the most simple transport mechanism across the membrane, right? Now let's come to a little more complex mechanism. We have discussed only diffusion across the membrane, and diffusion mechanism, simple diffusion is utilized by those molecules which are very small and which are lipid soluble, right? Carbon dioxide, like oxygen, right? Now we come to some other substances, for example, glucose, right? Glucose molecules. Or let's suppose there is sodium, right? This is an ion. Or there is calcium. If these substances have to cross the membrane, how they will cross? Now understand clearly. Sodium or calcium, they are having charges on their surface. They are ions, they are charged molecules. Because they are charged and polar, can they dissolve into lipid? No. If this cannot dissolve into lipid, they cannot cross the membrane by simple diffusion, right? In the same way, glucose is a larger molecule. Can it simply dissolve into membrane and cross? Answer is no. So now we have to think that these are some examples, some substances which have to cross the membrane, but these substances have a trouble. The trouble with these substances is that they cannot dissolve into lipid bilayer due to their larger molecular size or due to their polar nature or charge nature. So simple diffusion will not help. So how we can move these substances, right? Now for that purpose, in the cell membrane, there are special type of protein molecules, right? And those protein molecules facilitate the diffusion, right? How? For example, let me tell you about the glucose diffusion across the membrane. It cannot simply diffuse. Its diffusion of glucose from high concentration to the low concentration cannot be a simple process because it cannot cross the membrane. We have to facilitate this diffusion by some special mechanism. What is that special mechanism? You must be knowing that insulin, have you heard of insulin? Insulin stimulates the cell and then glucose entry into cell increases. It means insulin increases the uptake of glucose by the cells. Who knows the mechanism? How insulin increases the uptake of glucose by the cells? Anyone? Okay, insulin will come here and say, hey cell, please take glucose, I'm here, I request you. What insulin will do? Yeah. Insulin has receptors, then
change the configuration of something, open the channel.
yeah, something will happen. Let me tell you with little detail. Big detail we'll study in videos of receptors. Actually, let's suppose, first of all, let's suppose this is the molecule of insulin, right? Insulin is a large peptide molecule. It is made of a lot of amino acids. Can insulin go into cell? No, it's a large molecule, right? And so what will happen? Insulin will bind with the receptor on the surface of the cell. On the surface of the cell, right? Now this is insulin receptor. You will discuss insulin receptor and its mechanisms in detail in the lectures and receptors. Here I'm giving you a very brief example that insulin come to a target cell. Cells which are responsive to insulin must express insulin receptors on their surface. Is that right? When insulin bind with the receptor, this insulin receptor has extracellular domain and yes, intracellular domain. When insulin bind with the extracellular domain, right, it alters the structure of this receptor, right, and intracellular domain of the receptor is activated. You will learn later that it undergoes autophosphorylation, but I'm just making it brief that intracellular part of the receptor, right, gives signals, multiple molecular signals, and these signals do many intracellular activities. One of the activity is that within the cell there are special type of vesicles. What are these? Membrane bound vesicles. Cells which are insulin responsive, they are having preformed intracellular membrane bound vesicles. In these vesicles, these are the membranes, right? Lipid bilayer membrane, and in them there are glucose channels. What are these? Glucose channels. So what is happening that cells which are responsive to insulin, these cells are having intracellular vesicles, and these intracellular vesicles which are made of the membrane, they are having special protein channels, and these channels are basically channels for glucose. These channels allow the movement of glucose. Until these vesicles and their associated glucose channels, or we call them glucose transporters, until they are present within the cell, they cannot be utilized for the transport of glucose, right? And when insulin comes and binds with this receptor, it gives multiple intracellular signaling pathways, and these pathways will force these vesicles to move towards the surface. And when these vesicles will travel towards the surface, what will happen? These vesicles will touch the surface here. Is that right? And then vesicular membrane and cell membrane will fuse with each other, right? And these are your glucose transporters. And what will happen when multiple vesicles move towards the cell membrane, fuse with the cell membrane expand there, and suppose this is now vesicular membrane. This is a patch of membrane which is from the vesicles. This is a patch of the membrane. Actually, these are three vesicles fused here and here. And of course, they were having what? Glucose transporters. So what insulin has done? That insulin bound with its receptor, and insulin receptor initiated intracellular molecular events which transported the glucose transporters to the cell membrane, and immediately cell membrane permeability for the glucose is increased. Now glucose can permeate through the membrane because now membrane is having glucose transporter proteins. Is that right? And now glucose will be able to cross, and of course glucose will move from high concentration to low concentration. If you have eaten a lot of carbohydrates and glucose level in your blood is high, then insulin level will go up and they will go to the different cells like liver cells, muscle cells, fat cells, and their insulin receptors will be activated, and then intracellular glucose transporters will appear on the surface, and parts of the membrane become highly permeable to glucose, and now glucose will move again from high concentration to low concentration. Because glucose is moving from high concentration to the low concentration, this mechanism is still called diffusion. But this diffusion of glucose has been facilitated by the presence of glucose transporter proteins. So this type of diffusion across the membrane is called facilitated diffusion. That it has been facilitated by presence of certain protein molecules. Here those protein molecules are glucose transporters. So I will write it here. First of all, we discussed simple diffusion, and now we are talking about, yes, facilitated diffusion. What is facilitated diffusion? In facilitated diffusion, of course, substances diffuse from high concentration to low concentration, but because these substances are either polar or these substances are larger molecular weight, so they don't dissolve into in the lipid bilayer of the membrane. So presence of certain carrier protein or channels facilitate the movement of those substances, and such movement of substances from high concentration to low concentration is called, yes, facilitated diffusion. Let me tell you another example of facilitated diffusion. We were talking about calcium and sodium. Do you think sodium is present more inside the cell or outside the cells? Yes, sodium is more present outside the cells, and calcium is more present normally. Yes, calcium is also present in higher concentration outside the cells, right? Calcium, sodium and calcium are in higher concentration outside the cells, and potassium is in high concentration inside the cells. You can just think of that cells are the bag of potassium floating into sea of sodium and calcium. Is that right? Cells are the bag of potassium. Why sodium is more outside and potassium is more inside? We will learn after a few minutes that every cell has a special transporter, we will discuss later, which is called sodium potassium ATPase. These transporters are present in every cell membrane, and they continuously keep on throwing the sodium out of cell and accumulating, yes, potassium inside the cell. So due to the presence of sodium potassium ATPases in almost every cell membrane is throwing sodium out of the cell and accumulating potassium into the cell. Is that right? Now, as we know that sodium is in higher concentration, yes, outside the cell, and it is inside the cell in low concentration. If sodium needs to move from outside the cell to the inside of the cell, of course then sodium will be moving from higher concentration to low concentration. So it will be a type of diffusion. But we know that sodium cannot dissolve into lipid bilayer. So movement of sodium from high concentration to low concentration cannot be simple diffusion, because sodium cannot dissolve into lipid bilayer. So for this purpose, sodium diffusion from extracellular environment to the intracellular fluid has to be facilitated by some special mechanism. Nature has provided our cells with special type of channels, right? These channels act as sodium, right? Sodium channels. What are these channels? These channels are again special type of proteins, right? Let me make a channel here. Actually, let's suppose this is one peptide chain, right? This is another peptide chain. Here is another peptide chain, and now what happened? I'm showing in this diagram 1, 2, 3, 4, 5. These are 5 peptide chains. If they are put together, right? If they are clumped together, they will convert into a channel. They will convert into a channel, and this channel is made of multiple peptide chains. And in the channel, suppose 1, 2, 3, 4, and 5. These are the peptides which are put together, and they are now making a channel. In the center of the channel, there is water-filled tunnel, right? So it means this is a protein channel, and in the center of the channel there is water-filled tunnel, and now sodium can pass through the water. It cannot pass through the lipid, but it can pass through the water. So those cells which allow the sodium to move across the membrane, or those cells which allow the calcium to move across the membrane, or those cells which allow the potassium to move across the membrane, they are having sodium or calcium or potassium channels, right? Now let's suppose we put here a sodium channel. Now, in this sodium channel, what is happening? It will allow the movement of sodium from extracellular environment to the intracellular environment. Is that right? So it means the presence of these channels in active state facilitate the movement of sodium from high concentration to low concentration. So movement of sodium through the sodium channels is an example of facilitated diffusion. This is another example of facilitated diffusion, right? Here it's worth mentioning that different channels are operated in a different way. Some channels are open all the time. For example, every cell has some potassium channels, and these potassium channels are open all the time. They are unregulated channels, they are open all the time. And from every cell, some potassium all the time keep on leaking outward. These are called potassium leaky channel. What are they called? Potassium leaky channel. So some channels, like potassium leaky channels, they remain open all the time, right? Opposite to that, there are other channels which are well regulated by special mechanism. They only open under special circumstances. For example, there are voltage gated sodium channels. There are some sodium channels that normally cells are having resting membrane potential. You will study it later, that when cells are resting, many cells are electrically negative minus 90 millivolt inside as compared to the outside, right? We call it resting membrane potential. Suppose normal cell has resting membrane potential of minus 90 millivolt. Many sodium channels at this potential remain closed. These sodium channels remain, yes, closed. They are having a special voltage sensitive gates. These gates are not made of iron, they are also peptides. There is one gate outside, inside we'll discuss in higher classes that thing, but many sodium channels are having special peptide gates, and these gates are voltage sensitive. At minus 90 they are closed. But if you alter the potential within the cell, rather than minus 90 millivolt, if you make it minus 60 millivolt, at this voltage, this protein moves away. And if this peptide component moves away, right, channel will open. Such channels which are sensitive to the voltage changes and they open at a specific voltage or they close at a specific voltage, Such channels are called voltage gated channels. So some of the sodium channels are voltage gated channels. So at a very specific voltage, voltage gated sodium channels may open and then part of the membrane become highly permeable to sodium, and then sodium will move from high concentration to the low concentration, right? And that will be an example of what? Facilitated diffusion. Am I really clear? I think right now it's worth mentioning there are other types of sodium channels also. These different types of sodium channels are present in different tissues. There is another type of sodium channel. They are not regulated by the voltage. Rather, these are funny. They are having a peptide here. Right? Now, this particular point is not sensitive to voltage. This is sensitive to some neurotransmitter or hormone. For example, if acetyl, have you heard of acetylcholine? Acetylcholine? When acetylcholine comes and bind with the sodium channel, this channel will open. Are you understanding? This channel will open, right? So there are some sodium channels which are not voltage gated channels. They are gated by or controlled by or sensitive to certain specific binding substance. This binding substance may be neurotransmitter like acetylcholine, or it may be a hormone. Is that right? So such channels which open or close in the presence of a specific neurotransmitter or in the presence of a specific hormone, right? Such channels are called, yes, they are not called voltage gated channels, they are called ligand operated channels. Ligand mean a specific substance which bind to a specific protein. So acetylcholine is ligand, right? If some other substance bind here, that is ligand for this protein molecule or channel. So such channels are called, yes, ligand operated channel. Ligand gated channels or ligand operated channels, right? So as you will study at advanced level medical sciences, you will come to know there are many channels in the membrane, right? And they are altering their function under different stimulus. Some are like potassium leaky channels. They are unregulated channels. They are open all the time. Then there are voltage sensitive or voltage gated sodium channels, as well as there are voltage gated calcium channels. There are voltage gated potassium channels also. So there can be potassium leaky channels as well as there can be potassium voltage gated, right? In the same way, there can be ligand gated channels. Ligand means any specific neurotransmitter or any specific hormone or any other substance which specifically bind with a specific protein, right? So ligand operated channels, some of the sodium channels are ligand operated, some of the potassium channels are ligand operated, some of the calcium channels are ligand operated. Is that right? But all these channels, either they are voltage operated or ligand operated or they are leaky, all of them, when they are active, they increase the membrane permeability to a specific ion and let the ion diffuse across the membrane. And all of them are examples of what type of diffusion? Is it simple diffusion? No. What is this diffusion? Facilitated diffusion. Is that right? So what we learned up to now, that facilitated diffusion is an example of protein mediated cell membrane transport mechanism, because these are proteins. So glucose transport was also an example of facilitated diffusion. Ions transport across the membrane is also an example of facilitated diffusion. Do you have any question here? There is no question. Now, let's go to another transport mechanism if you don't have any question up to this. Up to now we have discussed simple diffusion, where substances are moving from high concentration to low concentration, and simple diffusion does not require any type of energy input. Do you think we need energy input to move the substances downhill? No. When substances are moving from high concentration to low concentration, we say substances are moving downhill. So simple diffusion does not require any input of energy. Then we talk about, yes, facilitated diffusion. In facilitated diffusion, again substances are moving from high concentration to low concentration, right? Only presence of special transporters or channel facilitates the diffusion. Is that right? Because substances are again moving from high concentration to the low concentration, so again we don't need to utilize some sort of energy. So it means simple diffusion and facilitated diffusion can occur across the membrane from high concentration of the substances to the low concentration of the substances without any input of energy, and such transportation mechanism in which energy is not invested, we call them passive transport. So these two transport mechanisms are what type of transport mechanisms? Yes, please. Passive transport mechanisms. Do you have any question up to this? Yes, please. Is there any question? There is no. Now we'll come to another type of transport across the cell membrane, in which we will utilize energy. As we discussed earlier, that extracellular environment is rich in sodium, and intracellular environment is rich in potassium. Is that right? And we discussed why it happens. I was telling you that every cell membrane is rich in special type of transporter proteins, and these proteins are called, yes, sodium potassium ATPases. Now what is the speciality of these proteins? These proteins are special type of carrier proteins, right? They are having 3 pockets here, and they are having 2 pockets here. Now, in this pocket, it will bind sodium, and sodium. And here it will bind, yes please, potassium. So what really happens, this is a very special type of protein which is inserted in the membranes of all cell, right? What is the special point related with this protein? That it has three domains on one side and 2 domains on other side. The 3 domains are usually hanging inside the cell, and they love to bind sodium. They love to bind sodium. And these domains which are hanging outside the cell, they love to bind, yes, potassium. These are potassium binding domains. Usually it binds to potassium, and these 3 are, yes, sodium binding domains. Usually they bind 3 molecules of sodium. When this molecule is loaded like this, it opens a special pocket here. And in this pocket, once this is loaded, it opens a special pocket, and in this pocket what will fit in? ATP molecule. Now, this domain of the molecule has capability to break down the ATP into ADP and phosphate, and the molecule will be energized when you break down the ATP into ADP and phosphate. So what really happens, we can say it will bind ATP and release out ADP and inorganic phosphate. And of course, during this process, what will be released? Energy will be released by the breakdown of ATP. That energy will be utilized by this special molecular motor or special protein molecule. Under the influence of that energy, this will flip And when it will flip, it will move like this, twist like this. So what will happen? Sodium domains will go out and potassium domains will come, yes, in. So the new configuration of the molecule will be like this, that now sodium domains are outside and potassium domains have flipped inside. As soon as sodium domains are outside, they open and they release sodium. So 3 sodium will be transferred from intracellular environment and pumped into extracellular environment. And these domains will also open, and what will come out? 2 potassiums, right? So what really happened? This is a very special type of integral protein in the cell membranes. It is so universally present that this protein is present in every cell membrane. Is that right? What is the function of it? That it is responsible to take the sodium, pump the sodium out of the cell and pump the potassium into cell. We already know the sodium concentration outside the cell is very high, and we know that potassium concentration is already present inside the cell in high concentration. So what does it mean? That this molecule has to pump the sodium from low concentration to high concentration. So it is pumping the molecules downhill or uphill? Uphill. And if you want to pull anything uphill, you need to invest energy. You are experienced, right? So this molecule or this transporter is transporting the ions against the concentration gradient, that sodium from intracellular environment is continuously being pumped to extracellular environment from low sodium concentration to high sodium concentration. At the same time, when 3 sodiums are pumped out, 2 potassiums are pumped from extracellular environment to the intracellular environment, again against the concentration gradient of potassium. So we can say that this sodium potassium pump, or we call it sodium potassium ATPase, because it has capability to break down the ATP. So we call this enzyme, this component is enzymic. So we call it, this is working like an enzyme to break the ATP. So we also call it sodium potassium ATPase, right? So this sodium potassium ATPase or sodium potassium transporter is present in every cell and continuously throwing the sodium out of the cell and pumping the potassium into the cell, so that it is responsible to maintain the high extracellular sodium concentration and high intracellular potassium concentration. Is that right? Am I clear? And these sodium potassium ATPases, as I told you, they are present in every cell. They utilize a large portion of your ATP. Is that right? So this is one example. Now, because substances are moved against the concentration gradient, so this is an example of active transport. It cannot be passive transport. And secondly, in any transport mechanism, when energy is utilized, that transport mechanism is said to be active transport. You know, simple diffusion was passive transport. Facilitated diffusion was passive transport. Because in simple diffusion and facilitated diffusion, substances are moving from high concentration to low concentration. But when we are talking about example of sodium potassium ATPases, they are moving the ions against the concentration gradient, right? So they have to utilize energy in the form of ATP. So sodium potassium ATPase and transfer of sodium to the outside of the cell and potassium into the cell is an example of active transport. Now, in this active transport mechanism, we are using the ATP directly at the transport site. ATP energy is utilized directly at the transport molecule or transport site. So we say input of energy is in, there is a direct input of energy in transporting mechanism. And whenever biologically energy is directly utilized in transporting mechanisms or transporting proteins, we say that this is primary active transport. Again, let me tell you. Whenever we move substances against the electrochemical gradient, we say there is a mechanism of active transport. In active transport, we need to use energy. If energy is utilized directly at the site of transport, then we say there is primary active transport. So this is not only an active transport, but energy is utilized at the site of transportation activity. So this should be called what type of active transport? Yes, primary active transport. Is that right? Any question up to this? Another example of primary active transport is, you may be knowing that within the cells there is a network of tubes which is called endoplasmic reticulum. Now, if there is a lot of calcium in the cytoplasm, calcium should be pumped actively into endoplasmic reticulum. Endoplasmic reticulum is a network of tubes, right, within the cell cytoplasm, right? And actually they are very rich. These tubes are full of calcium. Whenever calcium goes out of endoplasmic reticulum, this calcium should be pumped back. And if calcium has to be pumped from cytosol back to endoplasmic reticulum, calcium is moving against the concentration gradient. So it has to be passive transport or active transport? Active transport. And there is a calcium transporter here. There is a pump here which loves to move the calcium. And this pump, whenever it moves the calcium, it breaks down the ATP into ADP and inorganic phosphate. So this is also having an enzyme activity of ATPase. Now this calcium transporter is pumping the calcium from cytosol to the endoplasmic reticulum, from low concentration of calcium to the high concentration of the calcium. So transportation of calcium is against the concentration gradient, so it must be considered active transport. And in this active transport, because energy, biological energy is utilized by the transporting molecules directly, so it should be called primary active transport. And what should be the name of this protein? Calcium ATPase, as this was sodium potassium ATPase. Is that right? What is this? This calcium ATPase. Is that right? So these are two examples of what type of transport? Primary active transport. I think now it's important that we should talk about secondary active transport. Anyone is confused up to this? No one is confused? Anyone sleepy? Okay. Now we talk about what is secondary active transport. In secondary active transport, substances are transported across the membrane against the concentration gradient, but energy is not utilized directly at that very point. There is indirect utilization of energy. I think you don't understand anything. Let me take a help of a diagram. Then what is secondary active transport? Let's suppose there is lot of glucose here. Glucose molecules are here. We need to move these glucose molecules from this point up to this point, right? Okay, I will give you a very simple example. Let's suppose this is your gastrointestinal system, GIT. It is your GIT, not anything else, please don't get confused. This is a cell, right? You have taken some Coca-Cola and I hope a lot of glucose has gone in, right? Now you want to take all this glucose inside the cell and you don't want to leave any glucose in the lumen. So when this cell will keep on taking up the glucose concentration in the cell may become high, but still it should keep on taking up the glucose. It means in the end it is taking the glucose against the concentration gradient. It means glucose eventually need to be pumped against the concentration gradient. Is that right? Now how it really happen? Let's suppose this cell I put here. Actually in this cell, this is called luminal side of the membrane. This is on the lumen and this is the basal side of the cell. Here are your friends. Who are these? Sodium, potassium, ATPases. And these are the site of what activity? Primary active transport or secondary active transport? Primary active transport. Let me make it more clear. I'm making one cell of GAT here, right? This is luminal side and this is basolateral side, right? Of course, there are many cells, not one cell. Now, I said that on this side of the cell, what are these? Yes, please. Sodium, potassium, ATPases. They are taking the sodium against the concentration gradient outside and they are bringing the potassium inside. Is that right? Am I clear? The function of this sodium potassium ATPases is to keep the sodium concentration in the cell very high or very low? Low. And here sodium concentration very high. Is that right? Now we remove this cell at this level. Now this is your what is this? Luminal membrane, and this is your basolateral. And because this pump is working here, it is keeping the intracellular sodium level very low. Is that right? Now, from here, it has to take what in? Glucose. Now, there are very special type of transporters present on this side. I will draw these transporters here. These are very special type of transporters which are responsible to transport the glucose. Let me make one transporter big here. Let's suppose this transporter is here, right? Now this transporter protein has two pockets. In one pocket, it can bind glucose. In one pocket, it can bind glucose, and in other pocket, it can bind sodium. If only glucose bind, this transporter will not work. If only sodium bind, it does not work. This transporter protein can only be active when sodium binding domain and glucose binding domain, both of them are appropriately loaded. Is that right? If both of them are loaded, this protein will flip inside, right? Its next configuration will be like this. Right? So sodium will be released in, as well as, yes, glucose will be also released inside. Now what we have seen, that this is a protein which can transport sodium and glucose together. This is an example of co-transporter. This type of transporters are called co-transporter. It is sodium glucose co-transporter, or some people simply call such type of transporters symport. Symport or co-transporters. So there are many types of co-transporters. Here I have given an example of sodium and glucose co-transporter. Now this co-transporter can take the sodium and glucose from the extracellular environment and bring them to intracellular environment. Now in this example, sodium is going down the concentration gradient from high concentration to low, and glucose is moving, yes, from low concentration to high concentration. Even if you have accumulated lot of glucose here, still it will keep on pumping the glucose in. Now, glucose is moving from low concentration to high concentration because last molecule of glucose will be also sucked up. So is it an active transport or passive transport? Is it glucose is transported against the concentration gradient or with the concentration gradient? It is against the concentration gradient. Glucose is transported downhill or uphill? Uphill. So it is passive transport or active transport? Active transport. It is active transport of glucose. It is active transport of glucose. Is that right? But it is facilitated diffusion of sodium. Sodium is moving from high concentration to low concentration. So this concept is little bit complex, but I hope your complex mind will understand it, right? That same transporter is moving one substance down the concentration gradient and other substance up the concentration gradient. So the substance which is going down the concentration gradient is sodium, and presence of this carrier has facilitated the movement of diffusion of sodium from high concentration to low concentration. So we'll say sodium glucose co-transporter is transporting the sodium as an example of facilitated diffusion, and transporting the glucose an example of active transport. But do you think any direct energy is utilized here, ATP? No, because ATP is not utilized here, so it is not primary active transport. But from where the energy comes for transportation of glucose? Now this is a very important question, because glucose is being transported from low concentration to high concentration, so it is active transport, there must be some input of energy. But ATP is not directly utilized there, so it cannot be primary active transport. So what kind of transport is it? Let me tell you. Actually, this transporter will keep on working until inside the cell sodium concentration is low. Sodium gradient outside the cell should be high and sodium concentration inside the cell should be low. So this sodium gradient, right, is produced by utilization of ATP by this pump. This pump was, whatever sodium is coming in, this pump is actively throwing out. So this pump is utilizing the energy, sodium potassium ATPases are utilizing the energy to keep the intracellular sodium concentration very low, and when sodium diffuse from high concentration to low concentration, energy which is released is utilized by this transporter protein to drag the glucose from low concentration to high concentration. The classical example to prove that this mechanism is using the energy of this is that scientists made this channel, this transporter dysfunctional. There is a substance called obane, a toxic substance called obane. They exposed the cells to obane. Obane will especially made this part dysfunctional. And as soon as this cell will be exposed to the obane, sodium potassium ATPases stop working, stop utilizing the ATP, and intracellular sodium level will start going up. And when intracellular sodium level will go up, this sodium will not come in. And if this sodium cannot come in, this glucose cannot also come in. So this was an excellent proof that energy which is primarily used here, right, is actually utilized to keep the intracellular sodium down. This energy is indirectly responsible, right, to actively transport the glucose, right. So this sodium glucose transporter or co-transporter is moving the glucose from low concentration to high concentration as active transport mechanism, but this active transport mechanism is not having the biological energy input directly into this transport mechanism. Biological energy is utilized somewhere else. So it is having indirect energy utilization. So we call it secondary active transport mechanism. Am I clear? What is this? Secondary active transport mechanism. Another example of secondary active transport mechanism is that there are some other type of transporters present here. On one side they can bind sodium, and on other pocket they can bind amino acids. And when both of them are loaded, it will flip in. Sodium will go down the concentration gradient and amino acid will go up the concentration gradient. So it means in the gastrointestinal system, sodium and glucose co-transportation or sodium and amino acid co-transportation are example of secondary active transport. Am I clear? Any question up to this? So is there any, you are clear what is simple diffusion? Simple diffusion does not utilize any proteins. Facilitated diffusion is from high concentration to low concentration, but utilization of special protein, but no energy utilized. Right? In facilitated diffusion, there were carrier proteins, transporters, or there were channels. Glucose transporters were there and ion channels were there. Then we were talking about primary active transporters, where energy is utilized in moving the substances or molecules against the concentration gradient. Is that right? And in primary active transport, the transporting mechanisms are utilizing the ATP directly at the site of transporting mechanism. In secondary active transport, substances are moved from low concentration to high concentration, but they are transported in association with another molecule which is going down the concentration gradient. Is that right? The classical example of secondary active transport is glucose, sodium co-transportation, or sodium and amino acid co-transportation. There are some other cells in the body, they also utilize similar mechanism. I will show you those cells are having some transporter, and on one side these transporter can bind sodium, but inside the cell they can bind proton. Now listen carefully. This is another transporter, but it is different than those two. These transporters were having sodium and glucose. Both of them were taken from outside and transported inside. So this was an example of co-transportation. But this transporter has two domains, one extracellular domain, other intracellular domain. Extracellular domain binds sodium, intracellular domain binds proton. And then sodium comes down its concentration gradient, right? And when it will flip, what will move out? What will move out? Proton will move out. And even if there is high concentration of proton outside, still this will keep on pumping the proton out until sodium can move in. This is another example of what type of transport? Protons are moved against the concentration gradient. So this is an example of active transport, but ATP is not directly utilized here. So what is this? Secondary active transport. Another important thing, can it be co-transport? No. In co-transport, two things should move in the same direction. Here the two molecules are moving in opposite direction. These are called counter transport. What is it called? This is an example of counter transport, or such molecules are called antiports. These co-transporters were symports. You remember, they were symports, and these are antiports. So secondary active transport mechanisms are those mechanisms in which two molecules are moved across the membrane and energy is utilized indirectly, right? If both molecules are moving in the same direction, then these mechanisms are called symports or co-transporters. If both molecules are moving in opposite direction, these are called counter transports or antiports. Am I really clear? Let's have a break. Right, so let's have a little review what we have done up to now. There's a test. If a substance is simply diffusing through the membrane, moving from high concentration to the low concentration, this is an example of simple diffusion. If a substance is moving from high concentration to the low concentration across the membrane, but for its movement it requires special type of protein, which may be channels or which may be carrier proteins, that is an example of facilitated diffusion. If a substance is moved across the membrane from low concentration to the high concentration, whenever substances are moved from low concentration to high concentration, they are moved against the concentration gradient, that will be an example of active transport. And if active transporter proteins are directly utilizing the energy, then that is an example of primary active transport. If substances are moved against the concentration gradient, right, but transporting mechanisms are not utilizing the energy directly, that is an example of secondary active transport. In secondary active transport, if two molecules are moving in the same direction, that is an example of co-transport or symports. If during the secondary active transport, two molecules are moving against the direction of each other through the membrane, that is an example of counter transport or antipode. No problem up to this? Now, what we have learned that simple diffusion does not require any carrier protein, right? So simple diffusion is not protein mediated transport, simple diffusion. But other example we discussed about, yes, what was that? Facilitated diffusion. And in facilitated diffusion, we needed the protein molecules. Plus, in case of primary active transport, in case of primary active transport, we also used protein molecules. And in case of secondary active transport, we also used protein molecules. It means these three mechanisms, facilitated diffusion or primary active transport or secondary active transport, all of them are protein mediated transportations across the membrane. Or they can also be said that they are carrier mediated transport mechanisms, carrier protein mediated transport mechanism. Now such transport mechanisms which use the proteins as carrier have some special specificities. For example, I told you there were the glucose transporters. You remember? This is an example of facilitated glucose transport. Or there were sodium and glucose transportation. Do you remember? That was secondary active glucose transportation. Now both of them are carrier mediated. Proteins are involved. Is that right? Now such type of transport mechanisms have some characteristics. One characteristic is that such transport mechanisms show stereospecificity. Now what is meant by this? Let me tell you. Actually these transport mechanisms are specific. If this is glucose transporter, right, it will transport only glucose, not amino acid. Even it will be so specific that glucose is of two types. There is D-glucose and there is L-glucose. You know there is D-glucose, which is dextrorotatory glucose, and there is L or levorotatory glucose. Actually, in our body, the glucose transporters are specific for D-glucose, and if L-glucose bind there, they will not transport. So it means these transport mechanisms are specific. If there is sodium glucose transporter, this same protein cannot transport sodium and amino acid. For the transportation of sodium and amino acid, we need a different carrier. Am I clear to all of you? So this concept that carrier proteins which are utilized in facilitated diffusion or primary active transport or secondary active transport, they display the phenomenon of stereospecificity, that they are specific. For example, sodium potassium ATPase does transfer the sodium and potassium, but it cannot transfer magnesium and calcium. So it's clear to you. This is one characteristic of carrier mediated transportation. Second is that these mechanisms are showing the phenomenon of saturation. I will tell you what is saturation. Phenomenon of saturation. Actually, let's suppose that in this membrane there are multiple glucose transporters. Now, if glucose concentration is very low, it will rapidly transfer. But if you keep on increasing the glucose concentration here, more and more carrier proteins will be utilized. And if you increase the glucose concentration very high, all the carrier will be saturated. Then further increase in glucose concentration will not further increase the transporting activity. Again, let me tell you. For example, if there is a special carrier protein here and the substance which is being transported by the carrier, if you keep on increasing the concentration of that particular substance, then with progressively increasing the concentration of the substance, there will be progressively increased utilization of the carrier. A time will come when concentration of substance may reach so high, all carriers are utilized. Then further increase in the concentration of substance will not further increase the transportation. Am I clear? We can show it by the graph. Let's suppose here we are increasing the concentration of glucose. Here we are showing the transport of glucose. Initially, as you are increasing the concentration of glucose, right, transport of glucose is progressively increasing. A time comes when all the transporters are saturated. When you further increase glucose concentration, transportation will not increase and it becomes straight line. So initially when you increase the concentration of substance, in the very beginning all the transporters are available. So there is very good transportation, rapid increase in transportation. But as you keep on increasing the concentration of the substance, a time comes when all the transporter become saturated, then further increase in concentration will not further increase the transport velocity. Am I clear? We say that at this point transport maximum has been reached. Am I clear? The classical example of this is in your kidney. Let's suppose this is your blood circulatory system and this is one of the nephron of kidney. In the kidney there are cells here which have transport mechanisms for glucose. So glucose filter from your blood into this nephron tube, and from here these cells transport glucose back to the blood. So normally right now from blood glucose is filtering into this space and all the glucose is reabsorbed, and in your urine right now there is no glucose. But if I keep on increasing the glucose concentration in your blood, then glucose delivery here will become progressively more, and then more and more glucose will be reabsorbed. A time will come when glucose concentration in blood is very high and so much glucose is loaded here that all the transporters are fully saturated and extra glucose spills into urine. This is what happens in diabetes mellitus, that when you have relative deficiency of insulin and glucose cannot be utilized by the cells. So glucose levels in the blood goes very high and so much glucose come into nephrons. Then nephrons cannot reabsorb all the glucose. So extra glucose spilled into urine. Am I clear? No problem up to this. So these carrier mediated transport mechanisms which are facilitated diffusion of primary active transport or secondary active transport, all of them show certain phenomenon. Phenomenon number one is that these proteins show stereo specificity, that these carriers are specific for their transporting substance. Number two, that they show the phenomenon of saturation and transport maximum. Is that right? And third phenomenon is that they show the concept of competition. For example, glucose is being absorbed from here. Another molecule which is very similar to glucose is galactose. Right? In the gastrointestinal system, if you provide a lot of galactose, then these channels will start using the galactose and glucose absorption transport will be reduced. So we say galactose is competing with there's a competition between the galactose and glucose for the same transporter and carrier protein. Am I clear? Right? So again, let me repeat it, that facilitated diffusion, primary active transport and secondary active transport. These are carrier proteins mediated transport mechanisms. All these mechanisms are specific for a given substance to be transported, number one. Number two, as you keep on increasing the concentration of substance to be transported, then these mechanisms become fully saturated. Is that right? And then their further increase in transportation is not seen. Thirdly, that these proteins or carrier protein transportation mechanisms display the phenomenon of competition. That if they are transporting one substance, if other substance is very similar to one and other substance also is available, then between the first and the second substance there will be competition to use the carrier proteins for the transportation purpose. Am I clear? No problem up to this. Having said all of this, now I will come to another very special type and the last type of transportation mechanisms. And the last type of transportation mechanisms which I am going to discuss you, they are given a very special name, and that is called vesicular transportation. Let me tell you what is vesicular transportation. Transport mechanisms. Vesicular transport mechanisms. In these mechanisms, what really happens that substances are transferred across the membrane by utilizing special membrane bound vesicles, right? For example, let me tell you, membrane bound vesicles are that this is lipid bilayer membrane and there is substance which is present in. This substance may be transported from outside to inside by making the formation of vesicle, or this may be transported from inside to outside. So I will discuss this. When through the vesicular mechanisms cells are taking up extracellular material inside, we say this is endocytosis. What is this mechanism called? Endocytosis. If cells make vesicles and capture extracellular material and take it in, we say there is a phenomenon of endocytosis. And if there is some substance which is present within the cell, that substance is loaded in a vesicle, then vesicle come and fuse with the membrane and throw the substance out, then that is called? Yes, very good. Exocytosis. Now let me go into little more detail. There are three types of endocytosis. One type of endocytosis is very common. In many cells, their membranes make small indentations, and then these indentations become deeper, and then they become more and more deep, and then this detaches. And in this small indentation or depression, just a little amount of extracellular water is captured with some soluble substances. So what is here? Only little extracellular water with some soluble substances. There is no special particle captured. We say cell is drinking extracellular water. Cell is drinking extracellular water. So what happens? In many cells, normally cell membranes make a small depression, and in that depression some small amount of extracellular fluid is trapped, and then membrane flows around that, make a vesicle which moves away, and some extracellular fluid is captured in the vesicle and taken into cell, transported from extracellular environment into intracellular environment, and in that extracellular solution there may be some proteins which are present in dissolved state, but there is no particle, not well defined particulate matter. This type of endocytosis is called, yes please, very good, this type of endocytosis is called pinocytosis. So what is pinocytosis? This is a special type of endocytosis in which cell is drinking extracellular fluid and whatever is dissolved in the fluid. Is that right? Then there is another type of endocytosis in which cell will trap a special type of particle. Let's suppose there is a bacteria here. There is a bacteria here, and cell membranes, especially this is displayed by the white blood cells, neutrophils, the macrophages. Cell membranes move around this bacteria. You are understanding me? Right? And then the cell membranes move around the bacteria and then fuse at that end. Progressively move to each other and eventually they fuse here and here. Right? And then this point is pinched off. And now this vesicle, along with the unfortunate, unhappy bacteria, is trapped in. This is not drinking, it is eating, because a special particulate matter has been taken. Special particle may be bacteria, it may be some fungus, it may be some small foreign body. This particle may be some piece of a dead cell, or this particle may be just carbon particle. This process is not drinking, this process is called eating by the cell, and this is called, oh yeah, I think all of you are interested in eating. All of you know this is called phagocytosis. The special thing in phagocytosis is that, to capture bacteria or particle is not easy, and I think this vesicle becomes really very heavy. To move this and pull it in, special type of protein molecules make polymerase. These are special protein molecules which are specially arranged around it to pull it in. These protein molecules are called, yes, what are they called? These are called actin filaments, actin protein. So in the process of phagocytosis, the intracellular actin molecule reorganize themselves to pull that vesicle in. You have noted that in pinocytosis, there was no involvement of actin molecules, but in phagocytosis, there is rearrangement of the actin molecules. Is that right? Then I come to another type of endocytosis. For example, liver cells are of course in the liver, they are called hepatocytes, right? Liver cells love to take the cholesterol from the blood into their cytoplasm, and cholesterol is taken in the form of LDL. You know LDL, low density lipoprotein, they are rich in cholesterol. If this is a liver cell, then this cell should express special type of, oh my god, this color is very bad, it's difficult to do. Okay, determination can do anything. This is a receptor for LDL. What are these? This is LDL receptor. LDL is the cargo for LDL transportation, and this is, what is this? LDL molecule. What is it? LDL, low density lipoprotein molecule. Now, in the liver cell membrane, right, within the membrane there are special areas which are expressing high concentration of LDL receptors. When LDL receptors become loaded with the LDL, then these receptors give signals inside the cell. And when they give the signals inside the cell, a special type of protein molecules come and bind to this part of membrane. These protein molecules, they are like pinching machines. These are pinching molecules. They pinch the piece of membrane and pull it in. Because membrane will be pulled in, so what really happens, that you will find that this piece of the membrane has become like this. These were receptors for Yes, LDL, and here is LDL. And now what were these? These receptors are loaded in area. As soon as the receptor become loaded, special proteins appear and bind under the surface of this membrane to the cytoplasmic phase. These proteins are called calatherins. Calatherins, right? These proteins move like molecular motors and drag the piece of membrane inside, right? These are called calatherine coated pit, PIT, calatherine coated pits, right? These calatherine coated pits progressively move inside. Eventually they convert into calatherine coated what? Vesicles, calatherine coated vesicles. And in this vesicles, what is trapped? LDL molecule along with their receptors, right? And calatherine will keep on pulling them in until these 2 points will fuse with each other, and then a very special type of enzyme will come and pinch it off, and this will move in. And now this has become calatherine coated vesicle, of course, in the cytoplasm, right? And now, of course, your friends are trapped over here. The name of your friend is LDL. And then what will happen? There are some other vesicles which are loaded with enzyme. These enzyme loaded vesicles, these enzymes are having special cutters. These vesicle and this vesicle will fuse together, and these enzymes will remove the LDL and remove the cholesterol. And what are these? Receptors will go back to bring more LDL in. So this is an example where LDL is being endocytosed, right? But this endocytosis of LDL is dependent on the presence of its receptors. So this is called receptor mediated endocytosis. Receptor mediated endocytosis, right? And this endocytosis is classically having involvement of calatherins. So how many endocytoses? There is 3 types of endocytosis. One is pinocytosis. It is not dependent on actin and it is not dependent on calatherine. Then there is phagocytosis, which is dependent on rearrangement of actin. Then there is receptor mediated endocytosis, which is classically dependent on calatherine. Is there any problem? There is no problem. Now we come to, this was the vesicular mediated transport of substances from the extracellular environment to the intracellular area. Is that right? Which is called endocytosis. Now, a few words about exocytosis. Exocytosis is displayed when cell has synthesized and stored a substance within the vesicle. Let us suppose this cell has synthesized a hormone. Beta cell of pancreas synthesize insulin, right? And suppose these are insulin molecules. Let's suppose these are insulin molecules, and these insulin molecules are stored into this vesicle. Is that right? When beta cells of pancreas are stimulated, then calcium influx occurs. Special type of calcium channel open and calcium comes in. Beta cells of pancreas are stimulated, calcium influx occurs, and calcium mediated or calcium dependent exocytosis start. Because when calcium come in, calcium interact with certain proteins here. These are also calatherins, and these vesicles will move towards the, yes, they will move towards the surface, right? And what will jump out? Insulin. So this is an example where there is exocytosis of intracellular, intravesicular substances, right? Here what we have released, there is calcium dependent exocytosis releasing insulin. So many endocrine glands, when they synthesize and store the hormones, hormones are released by the process of exocytosis, and this exocytosis is usually calcium dependent. Another example of calcium dependent exocytosis is release of neurotransmitters. For example, this is one neuron and this is another neuron. For example, this neuron has vesicle with acetylcholine. It is having vesicle loaded with acetylcholine. When calcium will go in, these vesicles will move to the surface, fuse here, and they will release which substance? Acetylcholine. So this is an example of calcium dependent exocytosis of neurotransmitter. Maybe depending upon if this neuron was storing epinephrine or norepinephrine or serotonin or dopamine or histamine or acetylcholine, whatever it was storing, calcium dependent exocytosis will lead to the release of its neurotransmitter substance. So in your central nervous system, neurotransmitters are released from the nerve endings by the process of exocytosis. From many endocrine glands, secretions are released into blood by the process of exocytosis. Is that clear? So these were all different type of transport mechanisms. I will just take a quick test and then we'll finish the class. Sodium is being transported against the concentration gradient. It's active transport, passive transport. Sodium is being transported across the membrane against the concentration gradient. It is active transport. Transporter is using energy directly. Primary active transport. Sodium is being transported across the membrane, right? Across the membrane along the concentration gradient through its channel. This is an example of facilitated diffusion. Glucose is transported across the membrane through its transporters along the concentration gradient. Glucose is moving across the membrane along the concentration gradient through glucose transporter. This is an example of, yes, facilitated transport of glucose, diffusion of glucose. Glucose is being transported across the membrane along with the sodium against the concentration gradient, and energy is not directly utilized. This is secondary active transport for the glucose. Cell is making some vesicles on the surface, and vesicles are going from the cell membrane to the cytoplasm. No reorganization of actin, no clathrin involved. Pinocytosis. Cell membrane is making vesicles and they are moving from the surface to the cytoplasm and they are loaded with the clathrins. Receptor mediated endocytosis. From cell membrane, vesicles are pinching off, going to the cytosol, cytoplasm, and there is reorganization of actin involved. Process is phagocytosis, right? Some vesicles from the cytoplasm move towards the cell membrane and fuse there and release their content in extracellular environment. Process is exocytosis. Is that right? Some substance are moving across the membrane along the concentration gradient. Along the concentration gradient. No energy involved, no transporter involved. Simple diffusion. And lecture simply finishes. Class dismissed.